And thanks for the invitation to ProImmune. Thanks to Beatrice as well for the introduction, but I think the concept of resistance to immune checkpoint is really bubbling at the moment, and there are many different reasons and ways that immune checkpoint are not working in some patients. But I think it's well approved that accepted that inflamed cancer can tend to respond really well to immune checkpoint, where those cold tumors are really resistant. However, there's an intermediate phenotype in patients, which is this excluded or immunosuppressed phenotype, and it's usually due to the expression of some immunosuppressive cells, such as MDSC. And at KY we're looking at targeting the different aspects. We've got some cytokine here that aim to bring new immune cells. We've got programs that target MDSC, but today I just wanted to talk about one of our programs, which is the aim is to target TRegs and deplete these cells in the tumor microenvironment. TRegs affect the immune response in the tumor by many different ways. Obviously, they are really high expressor of CD25, and that constitutes a sink for IL-2. They can express some immunosuppressive cytokine, TGF-beta and IL-10, for example. They can obviously kill directly your effector cells, but they can also decrease the presentation by decreasing the expression through, for example, CTLA4 binding to decrease CD80 and CD86, so another mechanism of action. So the problem with Treg, there's not really any specific marker that's only expressed on Tregs. Obviously, CD25 is highly expressed on those cells, CTLA4 as well. Another one of these markers is ICOS, and the advantage of ICOS, it's not expressed in naive cells. It needs to be induced upon activation, and it's from the same family as CD28 and CTLA4, but unlike CD28, it's not expressed on those naive cells. And upon activation, ICOS can then engage its ligand, ICOS ligand, and that's basically regulate the survival of these cells, and also the expression of pro and anti-inflammatory cytokines. And that really depends on which cell express ICOS. However, it's known, as been shown by people, and I'm going to show you some of my in-house data, that regulatory T cells, especially in the tumor microenvironment, are really high expressor of that target, and therefore consider potential targeting to deplete those cells. There's a lot of biology around ICOS as well in its role in B cell development, and that's mainly derived from all the knockout mice that were done around 2000 and published. And basically it's important for the formation of the germinal center and also the class switching of IgGs. This is some in-house data just looking at this differential expression of ICOS in different tissues. So here it's in a preclinical model, a CD26 model, where we looked at the expression in spleen and tumors. And when we look at the percentage of cells expressing ICOS, we can see that overall the spleen are less positive than, there's a lower percentage of positive cells than in the tumor. And there's an interesting profile as well, is that your CD8 and your CD4 non-Tregs and CD4 Tregs are having different percentage of positivity within this model, the Tregs being 100% ICOS positive. But it's not only about the percentage of cells that are positive, it's also about the relative expression of ICOS on these cells. And again, if you compare the spleen, which is quite low, compared to the tumor, but against the idea that ICOS in the tumor microenvironment is highly expressed. But then we look at the differential expression between the cells. Obviously, we don't want to touch those cells too much. So we also demonstrate here that ICOS expression in CD8 and CD4 is much lower than in Tregs, and just invite you to look at the axis that is broken here. And therefore, can we exploit this differential expression in a therapeutic way? That's not only valid for mouse. This is an example of just looking at the expression of ICOS in the blood of an healthy donor. And again, you've got your Tregs that are highly positive, both at the percentage and at the relative expression. So, it's something that, and I'm going to show you some data in patient tumor at the end of the talk. So what we want to achieve is this antibody with a dual mechanism of action. So the idea is we have those TRegs that express high level of ICOS that inhibit your effector cells. And the idea, we want to have an antibody with an IgG1 format, so an effector-enabled antibody that can engage NK cells or macrophages to kill by ADCC, ADCP. However, those cells will still bind the antibody, but because the level of expression is lower, and we know that based on model that the ADCC and ADCP is really dependent on the number of targets on the surface of your cells, this antibody can still bind to some of the target, and maybe through engagement of Fc gamma receptor 2b would give you some agonism as well on these cells. And the overall idea is that by decreasing Tregs and increasing the effector cell activation, you can get some anti-tumor response. So this is what we achieved, and we have this antibody called KY1044. Our platform, the Chi mouse, makes a fully human antibody, but we use a bit of a trick here. We use a Chi mouse that was already knocked out for ICOS. And the idea behind this strategy was to get an antibody that was cross-reactive to different species to not only facilitate our preclinical work, our pharmacology work, but also to have a good molecule to do all our tox work. And this is what we achieved. I will show you some data. So this antibody has dual mechanism of action. It can deplete Tregs and can stimulate effector cells. We've demonstrated that, and that's not something we really wanted, but it's actually happened. The antibody can bind to ICOS and block the binding of ICOS to its ligand. I'm going to show you some affinity data and some cross-reactivity data. This is a table showing it's SPR data looking at the affinity of the KD of the antibody. Actually, it's not the antibody, it's the Fab to do a one-to-one model with the SPR data. But the idea, we've got single-digit nanomolar for the three species, and the same for rat, which is not presented here. So a strong binder to all species. We validated the antibody in different in vitro assay, and in this example, this is looking at ADCC mechanism of action. And we're using two different assays here. The first one is a Promega reporter assay, so looking at the engagement of Fc gamma receptor 3. And we can see when we use ICOS expressing cells, so from different species here, and we use the antibody, we've got a strong engagement of Fc gamma receptor 3, which upregulate the luciferase, the reporter, with low nanomolar EC50, where when we use the isotype control, we don't have any signal. Obviously, this is extremely artificial, but it's a good way of screening antibodies. So we move into a more primary cell NK cell assay. So we isolated some NK cells from healthy donor, and we co-cultured them with ICOS positive cells. And you can see again, this is a totally irrelevant E2T ratio because it's 5:1 E2T ratio, but still we got a really strong killing potential with a picomolar kind of a, probably picomolar because of the E2T ratio, but it's really strong and potent killer of ICOS positive cells, so validating the first mechanism of action in vitro. But we also did a lot of work in looking at the agonism, and it's an example here using a primary T cells from healthy donors. So as I mentioned, those cells do not express ICOS unless you activate them in the presence of CD3, CD28. And when we activate them and then put the cells together with all that cocktail on a coated plate, so basically the idea we need to present the antibody properly, we get an upregulation of interferon gamma, and this is an example I think for 8 different donors. It's quite viable as you would expect for a primary cell assay, but it's still significantly upregulation. And we've got also upregulation of TNF-alpha, for example. We've done more work around looking at the agonism in solution. If you put the antibody in solution, it doesn't work. You need to really cluster this antibody to get agonism. And you also need to engage TCR with CD3, for example. But obviously the idea was to validate some of this mechanism in vivo, so we did a different experiment in syngeneic models. This is an experiment as a monotherapy here in a B-cell syngeneic model called A20, and basically we saw a really strong monotherapy response in this model, where all your control grow really quickly, but your tumors start growing, but then are rejected, and we've got the Kaplan-Meier on the right-hand side here. So really strong monotherapy efficacy. It has to be said that actually the monotherapy efficacy is not something you see in every model, far from that. It's more when you start combining with immune checkpoint blockers. And this is an experiment going back to the CT26 model. And we implanted the tumor subcut, and we dosed here, these 2 weeks of dosing. Every vertical bars represent a dosing day. And you can see that the control, isotype control, the animals have to be sacrificed really quickly. It's not really survival. We're dependent on home office regulation, as you know. But basically we had to kill the animal within 30 days. And both the KY1044, so just to mention something I forgot. In this experiment, we're using mouse IgG2A version, so it's not the human IgG1. And the idea is to have an effector-enabled antibody. When we look at the effect of KY1044 mouse IgG2A, we've got this small delay, but it was extremely reproducible. We've done this experiment around 10 times now. And you all the time have 1 or 2 animals that respond. You don't know why, but they respond, and again, that's reproducible. Similarly, PD-L1 in this anti-PD-L1 in this model that doesn't work. In purple, again, we've got a bit of a delay, but again, eventually the animal needs to be sacrificed based on their tumor size. So it's only when we combine both molecules that we got this strong synergism. And here in this experiment, we've got 70% of the animals that basically were totally cured of their tumor. And when I say cure, it's basically no sign of tumor measurable at day 60. And what we've done, we've took some of these animals, and as you would do in this type of experiment, we re-challenge them with the same cell line, so CD26, or with a new cell line, the MT6. And as you can see, so the re-challenge was done on that would be day 61, for example. When we re-challenged with CD26, we didn't see any growth during the time where normally we would see. However, the MT6, all the animals that were re-challenged with the MT6 show a sign of tumor growth. Again, suggesting that we've not only induced a strong anti-tumor immune response, but we also may have a memory response established to that tumor cell lines. So as a summary, so it doesn't work in every model. I think we have to be honest, and that's the case for every immune checkpoint blocker. But we've got some models where we see some efficacy, some that are more responsive than others. We've got some models that are not responding to anti-PD-L1 and yet respond to anti-ICOS. So again, it's possible that some of the patients that would be resistant to anti-PD-L1 could respond to anti-ICOS. The combination give us a really strong efficacy in certain of this model, and it's not, we see efficacy in PARP-C and BLAC6 model. But again, you've got some models that do not respond. The usual highly immunosuppressed model, the B16F10, is not responding to the monotherapy or the combination, and so is the ALK4 and this model, which is a breast cancer model that is quite strong in MDSC, is not responding either. So seeing if anti-tumor efficacy is great, especially the fact that we're using the actual drug candidate, at least on the Fab aspect of the molecule. But we wanted to look at the mechanism of action in vivo. So we went back to the CD26 model, and we implanted with tumors. The mice were dosed twice on days 13 and 15, and 24 hours after the second dose, the animal were culled, and we did some FACS analysis on spleen and tumor, and looking at different type of immune cells. And there's an example here. So we use the other control as well. So in the cell line, so it's quite variable, but you can clearly see the presence of TRegs in the tumor microenvironment. We're using this control, which is an anti-CTLA4 antibody 9H10, which has been reported in the literature and by others that it's a good depleting antibody for TRegs I invite you to look at the work of Sergio Quesada on this. But we use anti-ICOS in two different flavors. The Mars IgG2A, which is the strongest depleting format. We also use a Mars IgG1, which is the Mars IgG1 doesn't bind to Fc gamma receptor 4, the Mars, but it binds a bit to Fc gamma receptor 3, so it's not totally effector null. That's why you see a bit of depletion. But more importantly, the only one that gives us the strongest depletion of the TRegs was the Mars IgG2A version. Importantly, and based on, if you go back to one of the earlier slides that I've shown you, we didn't see any depletion in the spleen. And again, it's possible that is due to that different level of expression between the two tissues. When we look at the CD8 in this experiment, there was no much, there was nothing different between those and the control. The CTLA-4, and has been reported before, seems to upregulate a bit the percentage of T cells in the tumor microenvironment. But overall, what we were really keen to demonstrate was that this improvement of this ratio of CD8 to Tregs, and this ratio has been shown, high ratio has been shown to be a good prognosis for patients that would be treated with immune checkpoint blockers. And again, I want to remind you that in this model, CD26, the monotherapy doesn't work, or we got 1 or 2 animals that respond. Yet we can change the immune contexture and the tumor microenvironment in a way that we would hope help it to respond to immune checkpoints such as anti-PD-L1. We've done a bit more work around the PD. Instead of doing a single time point here, we've done a single dose, and we've monitored the tumor and the tumor microenvironment over a period of 8 days post dosing. So each time point we had some sampling there. And here we're looking at 3 different doses of 0.3 mg per kilogram, 3 and 10 mg per kilogram, dosed IP. And as you can see, even at a really low dose at 0.3 mg per kilogram, you've got already this strong depletion of the Treg, so extremely potent molecule. However, within 8 days post-treatment, at the low dose, you go back to your baseline quantity of Treg, so those cells are being replenished. However, when we maintain the dose at 3 or 10 during the course of the experiment, we maintain the percentage of Tregs really low. But what we've noticed is this is basically, again, that would be the previous data I've shown you at saline versus 10 mg per kilogram, and you've got this improvement of the ratio. However, we demonstrate that at intermediate dose, I mean, all of them are more than the control, but clearly the intermediate dose was showing a higher CD8 to Treg ratio, so there might be some work to do there. But what was interesting, when we took the tumors at endpoint at the end of the 8 days, and we look at the cells, for example, here CD8, but we've got the same data for CD4, we've got the same data for CD8, and we took those cells, we treat them with brifildin, and then did the intracellular staining for interferon gamma. All the cells that were there had the ability to express more ICOS. So at that stage, we don't know if this agonism or this activation is due to depletion of the Tregs or direct agonism of the cells. But clearly, again, not only you have a good microenvironment by increasing the ratio, but also those cells are quite activated. But with this in mind, with this kind of response in mind, we did more work and looking at different combination treatment dose. So we kept the anti-PD-L1 constant, but we played with the dose of KY1044 mice IgG2A. And you can see that we've got a good response with all of them, but clearly, again, the intermediate dose was giving us the strong response. So, we're trying to understand that more. We're doing some PK/PD modeling, and I'm happy to discuss a bit more about that. But it's a really interesting aspect. So obviously that's part of the preclinical work, and obviously we now full steam in terms of IND enabling activity. Finding the right indication and the right patient is at top of our mind, obviously. So we're spending a lot of time and efforts on the biomarker strategy and I'm going to show you a bit of a teaser here, but maybe next year I can come back with more data. But first, obviously you want the target to be expressed, so this is a joy plot of the TCGA. I'm really sorry, you probably can't see at the top, but we're looking at relative expression, and those are all the indication on the left-hand side. And clearly the indication express high level of ICOS. The top two would be thymoma and DLBCL, but also they have high level of ICOS expression. They're also growing in an environment where you've got a lot of immune cells, so they may also be quite in normal tissue, there's already high ICOS there. However, when you move to the next indication, you've got esophageal cancer, you've got stomach cancer, gastric cancer, you've got head and neck cancer. So quite a lot of interesting indications that have shown to respond to immunotherapy in the past. But obviously that's mRNA, and also there's been a lot of data published showing that there's a good correlation, not a lot, but there's been some data showing there's a good correlation between ICOS, mRNA, and protein expression. We wanted to really look at the level of expression at the protein level. So we developed our own IHC protocol, and this is growing, but at the moment we've already stained about 1,000 samples of cancer, focusing on 6 different indications. And the first thing we've shown clearly is that if you compare to the matching healthy tissue, you've got a really strong upregulation of ICOS positive cells in the tumor microenvironment. But we think it's not sufficient to just focusing on ICOS, so we're looking at other markers. So I'm not sure if you can see it. Maybe I'll. No, it's not going to work. So can you see? We've done a staining for FOXP3 and ICOS, so ICOS in purple and FOXP3 in brown. And this is an example of tonsils, so it's not cancer, but that was to develop the staining. But you can see that some of the cells are double positive and some are single positive. So we were quite happy with the staining. We did similar work looking at CD8 and ICOS expression, and you've got cells that are single positive and cells that are double positive in this tissue. So with this in mind, we also develop an algorithm to look at the expression, quantify the expression, the number of cells per millimeter square, and this is something we're doing at the moment. But just as an example here, I'm giving you an example of head and neck cancer. I think it's a great example because on the left-hand side you can clearly see, so be careful because the color are inverted, so is ICOS in brown and CD8 in purple, and you clearly see, and the quantification shows that your CD8 cells are not expressing high level of ICOS. They're probably ICOS, but the sensitivity of the IHC is not strong enough to pick it up. So again, demonstrating that this differential expression we see in preclinical and in the blood is also seen in cancer. The cells that are ICOS positive are not CD8 positive. However, when we move into the double staining of ICOS and FOXP3, you had, especially in head and neck cancer, you had a really strong co-expression of those two markers, again suggesting that there could be a really interesting indication and to deplete and change the ratio in those patients. So, hopefully I've demonstrated that we've generated using our platform, our KY1044 platform, and in which we had ICOS knockout. We generated an antibody that was cross-reactive to different species with a really strong potency, and it can induce ADCC on ICOS eye cells, and has some agonism property on effector cells. Because we had this cross-reactivity with mouse, we used that molecule to really demonstrate this strong anti-tumor efficacy, both as monotherapy and in combination with immune checkpoint blockers such as anti-PD-L1. And we also demonstrated some memory response. The pharmacodynamic work also validated what we saw in vitro. We see depletion of TRegs in the tumor microenvironment, even at really extreme, low dose, so extremely potent. But I think, more importantly, and that's where I get excited, we're starting really to look at the translational aspects. So we've stained 1,000 samples, and we've got now some really good indication in mind. We're spending a lot of time, and I can't present the data today, but around looking at signatures, so not only responsive signature, but also a potential predictive signature, and we're looking as well at immune profiling. So, the correlation between, again, it's a bit, we've talked about it today, but the correlation between the tumor microenvironment and the blood is something we're still looking at, but there's a lot of work ongoing, and we're looking at different cytokines that we've shown to be upregulated in the tumor microenvironment. So, with all this, I just wanted to thank you to stay around, and I want to thank the teams. It's been a lot of work from different people, and including in the discovery and in the development team. We hope to be in the clinic by January 2019. So thank you very much for your attention.